All right, hello everyone. Welcome back to TeleShadowing. My name is Amna, and today we'll be discussing a variety of different MCAT questions ranging from multiple disciplines. And Dr. Chima is here to provide insights on applying them to real-life cases and disease. So the MCAT is a big hurdle that many pre-medical students have to overcome in order to gain admission to medical schools in the United States. So today I'll just be going over a bunch of questions I picked out from the question bank called UWorld. We all love UWorld. And so I know that Minahil and a couple of our other med student friends have had panels on the MCAT and going over MCAT questions as well. But I figured that since I have now been through my MCAT journey, that I'd hop on here and provide my own few insights. So I handpicked questions from our friend UWorld, and these questions include topics that I could just go on and on about. But I know that a lot of us won't want to hear too much about organic chemistry, so I'll keep it relatively brief. So along the way, I'll give you sorts of tips and tricks and advice and like sort of mental frameworks on how I tackle these questions. And because some questions, they can be like long and convoluted and hard to digest, but when we break them down, once we chop them down to different pieces, into just the fundamentals of what they're asking, when we reframe the question to fit what we already know, like our knowledge bank, it makes it easier to just apply the basic concepts that we already know from our content review. So the most effective way, I'd say, to study for the MCAT and apply the content knowledge that you receive through books and through courses, if you choose to do so, is through practice questions. So I used UWorld, but there's plenty of other resources like different courses. So just pick the resource that works for you after doing your research. And not just doing questions is important, but reflecting and learning from your mistakes that you make. Because in fact, a lot of the questions that I've picked today, I actually got them wrong the first time I took them. But through reading the explanations and reviewing the supplementary materials and figuring out exactly why I got the question wrong, watching YouTube videos on the topics, reading my Kaplan books, I was able to nail down these concepts. And now I'm here. I'll teach them to you. So I was able to fill any and all content gaps that I had. So first up, we
Thank
have.
you so much. Amma, thank you so much. I really appreciate your time and giving us insight about your prep material and how you prepare for that. I really appreciate. And definitely when you will start doing your MCAT questions, I would like to correlate with our medicine cases. So this is how it will help you to be more passionate about learning MCAT, and you will see that whatever you are learning in biochemistry, biology, you are going to apply in med school and after residency, and then it will help you. So you will have a strong base, and on that, it will help you. So let's start now. Thank you.
Yeah, I totally agree. Definitely, it seems that when we're reading all this stuff, like learning about amino acids, the intricacies of glycolysis, gluconeogenesis, we don't understand the point of why we're doing it. But once we apply it to clinical situations, we'll see the big picture of why we're working so hard. So thank you for your insights. I'm sure we're all looking forward to it. So our first question I pulled out from the Biological and Biochemical Foundations of Living Systems. It's basically, we just call it BB, so bio and biochem. That's the third section of the MCAT. So our first question here.
I'm now pretty quick. I want to ask, can you please explain how many sections the MCAT and how much time we allowed for each block? And can you please give us a brief overview before we
Yes, of
start?
course. So the first section, we have chemical and physical foundations of living systems, or CP. It's mostly general chemistry, organic chemistry, physics, a little bit of physics, and some biochem can be intertwined in that. But for that, you have 59 questions and 95 minutes to answer them. So you've got passage-based questions, and then you also have discrete questions that don't come with a passage. You sort of have to read. They sometimes give you context in the question, stem, and then you have to apply your knowledge there. And then we've got passage-based questions. So after the first section, which is CP, chemistry, physics, you've got a 10-minute break. And then the CARS section, critical analysis and reasoning skills, where it's mainly about reading comprehension. You go through 9 passages, and each of them has 5 to 7 questions that you have to answer based on what you read in the passage and sort of make analyses on the author's purpose and argument and tone, stuff like that. After that, it's 53 questions total, 9 passages, 53 questions, and 90 minutes to do them. Then you have a 30-minute break, thankfully, because after all that reading, you need a snack and you need to take a break from the reading. Then you've got the biological and biochemical foundations of living systems, or BB. Mainly questions from biology and biochemistry, can incorporate some gen chem as well in there. You've got 59 questions and again 95 minutes to do them. And then you have another 10-minute break before the psychological and sociological, I believe, the biological and behavioral foundations of humans and living systems. So in that you hit a lot of topics on psychology, sociology, those kinds of theories, and then you also have the biological aspect of behaviors, which is like parts of the brain mostly. So like the amygdala, the how we react to certain situations and why, which part of our brain, some disease like Wernicke's aphasia, Broca's aphasia. So that all comes under the PS section, we call it, psych sociology. You've got 59 questions again and 95 minutes to answer them, and then you're done.
Thank you, Amna. Really appreciate.
Okay, so we'll go on to the first question in the BB section that I pulled out. So let me read it for you. Anyone wants to read it? I'll read it. It's fine. The enzyme phenylalanine hydroxylase catalyzes the conversion of phenylalanine to tyrosine. The disease phenylketonuria, or PKU, is caused by an inactivating mutation in the gene encoding phenylalanine hydroxylase. Which structure corresponds to the amino acid that will not form as a result of PKU? So this is what is called a pseudo-discrete question. So basically, I pulled this question out from a passage-based section. So it was with a passage, it came with a passage, but I believe that you don't exactly need information in the passage to be able to answer this question. You can easily do it without the passage. So that's what we call pseudo-discrete. It's kind of discrete. So first of all, what we need to do, we need to break down what they're telling us, because this is a lot of words, and it can be hard to digest at first. So we need to ask ourselves, what are they telling us and what do we need to find? So it's important here that we use our scratch paper that we're given to write out, like to mind map sort of what we have, what information we have, and what information we will need. So we'll write down something like this. So we know that the enzyme catalyzes the conversion of phenylalanine, which is an amino acid, to tyrosine. So we've got phenylalanine and tyrosine. Just small little notes like this can help you map out what you need to know. And then this enzyme, I just wrote the name here. So it's important that you use your resources for that reason. And we can also break it down even further to understand that phenylalanine hydroxylase is an enzyme that deals with adding a hydroxyl group, which is an OH group. So you can sort of keep that in mind when we're going through our answer choices. So we know that it's asking us what happens when the PKU, which deactivates the phenylalanine hydroxylase enzyme, what happens here? That means that we don't have any tyrosine. So now we need to understand what sort of prior knowledge we need to understand this question and how to answer it. So we've got our friends, the amino acids. So these are very, very high yield. I'd recommend memorizing, know the structures, knowing the ins and outs. This is the diagram I used here. My biochemistry professor provided it to us. So I'm not sure if it's on Google, but you can screenshot it here. So this just shows the properties and structure of each amino acid, but you'll also need to know the 3-letter codes and 1-letter code for each. So you can look them up there. There's plenty of resources out there. So to practice, I'd say just get a bunch of sheets of paper and just write them all down. Your typical paper and pencil. iPads are fine, but I believe that the traditional way of using paper and pencil is the best way to ingrain things in your mind. And also there's a lot of like apps to help keep your knowledge of these sharp. So once you've sort of nailed down the basics of all these amino acids, you can practice as much as you can through these apps. I used amino acid quiz, and I'd use it while I was like walking between classes. So notice the pKa's I've noted here too. These are important to note, and you'll need them in further breakdown of certain concepts that you'll find out while you're doing your content review. So we can go back to the question here. It's asking us which amino acid will not form if we've got this disease here. So we can see that tyrosine will not form, and we know that tyrosine is B. All right, so we know that the letter A corresponds to phenylalanine, letter C is tryptophan, and D is histidine. So don't let this overwhelm you. Once you've practiced and you've got all the amino acids down, you'll be able to recognize these as well as I can. And yeah, so basically what we did was we just took the question, we broke it down into simpler parts, we wrote down what was going on in our minds, and was able to lead us to the correct answer, and we were able to find tyrosine. So now Dr. Chima will probably talk a little bit more about the actual disease, so we can apply why we're learning all these things to really the clinical aspects of all this.
So Amna, can you please go over what's the right answer? Do you provide the options A, B, C, D, and then go over?
Yes, these were the options, and I went over them. Yes.
So you said this is the right answer, right? And
Yes.
did you mention why the other answers are not correct?
Yes.
Okay. All right, good. So what happens? So phenyl PKU or phenylketonuria is a genetic disorder that prevents the body from metabolizing phenylalanine. This is basically an amino acid, as mentioned, Amna mentioned, and found in the proteins. And if we left untreated, so PKU can cause a brain damage, it can cause some intellectual disabilities. So I'm trying to correlate this MCAT question clinically. So as I mentioned, phenylketonuria, also called PKU, is a rare inherited disorder that cause an amino acid called phenylalanine to build up in the body. So what happened? So PKU is caused by a change in the phenylalanine hydroxylase gene. And this gene basically helps create the enzyme needed to break down phenylalanine. And the pathophysiology of PKU is primarily attributed to the elevated levels of PHE and its metabolites, such as keto acid, phenyl pyruvate, and then there's degree levels of TYR amino acid. So they may also play an adverse role. Since this amino acid is an important precursor of three types of catecholamines or neurotransmitters like dopamine, norepinephrine, and adrenaline. So what happens when a patient has PKU? So symptoms happens basically is a metabolic disorder. So PKU is a metabolic disorder that can cause range of symptoms that can include neurological symptoms like seizure disorder, tremors. Patient can have a jerking movements in arms and legs. Patient can have some developmental delays like slow growth, taking longer for the pediatric population sit and crawl and walk, and their loss of interest in surroundings, and then some dermatological issues like rashes, eczema, like there's a lighter skin and hair color, then family members. And then there are the behavioral problems, like patient can have hyperactivity, patient can have show some temper tantrums, they can do self-harm, can cause anxiety. So these are all the behavioral problems. So, and then most importantly, the intellectual disabilities. It can a patient can have low IQ level and then can cause severe intellectual disabilities. And there are other issues like patient can have a musty disorder in breath and skin and urine samples, and more, then patient can have the microcephaly as well. So what's the treatment? So I will not go in detail, pretty quick. Basically, in adult population, what happens, adults with PKU may experience these complications. So if they don't follow a special diet that limits the foods and beverages that contain the phenylalanine, and so these include many protein-containing foods, and basically they have to have a lifetime dietary habit they need to change, and the lifetime diet with very limited intake of foods with phenylalanine. And these include many protein-containing foods, some artificial sweeteners like aspartame, and some other some medications and supplements. So healthcare provider and physician can refer to the dietitian to help the patients with that, and can help you to learn more about the specific dietary needs patient has. So it's very important to learn that PKU in adults find they function best while they're on the low protein diet. So basically, all these patients need to be on the low protein diet, and they should avoid high protein foods like milk, dairy products, meats, eggs, nuts, soy, and beans. So person with a PKU, an adult with PKU should avoid all these artificial sweeteners like aspartame we talk about, which contains the phenylalanine. So what can we eat, right? So that I mentioned, like dietitian can help us. We can refer the patient to the dietitian, or we can provide them the guide like medical foods, they're available. There are the shakes available. There's some powder design for PKU patients, and most fruits and fruit juices they can use, and most they can use non-starchy vegetables, and also they can use low protein breads and pastas. They can have fats like olive oil, butter, coconut oil, and sweeteners like sugar, white or brown sugar, honey, maple syrup. So, and small amounts of heavy cream. These are the things that they can use it. So this is how I want to correlate this MCAT question. So it will help you that why you are learning your undergrad level, all these basics concepts. So when you have a good base, so you can apply and you can learn more about it in medical school, and then you can learn more management, and you will see the more case studies, more patients during your residency, training years, and so you can help the patients after you get your license to be practiced. Let's move to our next case studies. Next,
Yeah, thank you, Dr. Shiva. I just wanted to point out how something so small like a tiny genetic mutation can affect this enzyme, which sets off just like a snowball effect, a ripple effect of all these different things. Like phenylketonuria is just caused by one small genetic mutation. So we should be grateful and thankful that all of our genes are working the way they should, how they're so intricately designed to in order to give us health and well-being. All right, we can go on to the next question I've got here. It's also from the BB section. So let me read it for you. Type 1 diabetes mellitus, or T1DM, differs from type 2 diabetes mellitus, or T2DM, in that affected patients stop producing insulin at a young age. Which of the following is most likely impaired in type 1 diabetes mellitus? All right, so this is another pseudo-discrete question, and it came with a passage, but we don't necessarily need the passage to answer the question. And when we come across these Roman numeral type questions, there's a couple of different strategies we could go about to frame this question and make it easier to get us to the right answer. So firstly, if we're able to immediately identify the correct Roman numeral or Roman numerals that could lead us to the right answer, that's one strategy, like knowing exactly what is the right answer. Another strategy is process of elimination, and these Roman numeral questions sort of make it easier to do that. Like for example, if you know that number 1 is wrong, then you can immediately eliminate A and C, and then you're only left with 2 options. So these Roman numeral type questions are pretty fun for me, especially to try and deduct just based on process of elimination and knowing your content. So sometimes it's a little bit unclear what the correct answer is at first, so process of elimination can help. So that was a little bit about strategy. Let's go on to the actual material. So the pancreas has plenty of functions in our body, both endocrine and exocrine. So endocrines like hormones that are circulated in our bloodstream, and exocrine such as like enzymes and certain things that our body needs to be able to function overall. And so cells with this paracrine function secrete substances that exert effects on neighboring cells. So what does that mean? So paracrine functions are when cells are able to deliver communication or signals or messengers or molecules to adjacent cells, and it's able to initiate this ripple effect of different different functions that these cells undertake. So the functional units of the endocrine pancreas, so the endocrine part of the pancreas which circulates certain hormones throughout our bloodstream, are the islets of Langerhans, which are made up of alpha, beta, and delta cells that secrete different hormones into the blood. So let's go, first of all, beta cells. Beta cells produce insulin, inhibits alpha, which inhibits alpha cells. So that means that beta cells exhibit paracrine function because their effects affect their functions affect the functions of their neighboring cells. So what are alpha cells? Why do they get inhibited by insulin? Alpha cells produce the hormone glucagon. So glucagon is the opposite of insulin. Insulin allows our cells to take in glucose from the bloodstream, and glucagon releases glucose from our cells into the bloodstream. And the final cell of the islets of Langerhans are delta cells, and they produce somatostatin. And somatostatin is a hormone that has a overall like inhibitory effect on digestive function, and it suppresses both insulin and glucagon release for certain reasons. And we can also see the exocrine function of pancreas, which secretes pancreatic enzymes. So the pancreas plays a big role in our body, of course. So let's go back to the question. The question states that type 1 diabetes mellitus is caused by a failure in insulin production. So we talked about insulin, how it allows glucose to enter cells from the bloodstream when we need energy to, because once glucose enters cells, glucose undergoes a lot of metabolic reactions that you've probably learned or you probably will be learning to give us ATP in the form of energy. So affected patients of type 1 diabetes mellitus most likely have impaired endocrine function, because we were talking about the endocrine functions are insulin, glucagon, and somatostatin. So if we got something wrong in our insulin, we don't produce insulin, that means there's an endocrine function. So we know that it should be either be 2 or 4, right? It can't be 1 or 3. So in this case, we've got a couple of different strategies. Again, if we know that it's not 1 or 3, we can eliminate answer choices A, C because it includes 1, and D because it includes 3. And then we're left with B. But if we want to go deeper, we will read the rest of the question, the rest of the answer choices, and see that what's the difference between 2 and 4? Why is 2 correct and not 4? Two says that the pancreatic beta cells are impaired, and number 4 says that pancreatic alpha cells are impaired. But remember, we said that the beta cells produce insulin, alpha cells produce glucagon. So that's just one thing that you'll just have to memorize. You can write it down, practice, use Anki. These small facts will help you in the long run. So in that case, we know that in type 1 diabetes mellitus, the impairment is in the endocrine function of pancreatic beta cells. Dr. Chima, would you like to expand more on diabetes mellitus?
Yes, sure. First of all, this question related to the anatomy and physiology we learned during the first, second year of medical school. So I would like to focus on what is pancreas. You can see in this diagram, so pancreas is basically a pear-shaped organ. It's located in the upper part of the abdomen behind the stomach. And basically it's about the size of our hand, and it helps with the digestion process. And it produces pancreatic juices called enzymes. They're the exocrine function of the pancreas, and then endocrine function related to the diabetes. As Amna mentioned, alpha cells, beta cells. So what happens? So basically this is anatomy and physiology. So pancreas has portions: head, neck, body, and tail. And posteriorly, there is an uncinate process. And then alpha beta cells is all the histology, that one branch of the anatomy we learn during our medical school. So pathophysiology, what happens with the pancreas? So normally the common conditions of pancreas happen, it can be the acute pancreatitis. So what happens? The inflammation of the pancreas. And symptoms: patient presents with the abdominal pain, patient can have a nausea and vomiting, they can have a fever, chills, and these are the common symptoms. Patient present with the acute pancreatitis. And so pancreatitis is basically the inflammation. And what happens? So this can lead to the malnutrition, because pancreas release the digestive enzymes, right? So when there's inflammation, so this means it's not producing enough enzymes that are needed for the digestive process. And then when chronic pancreatitis happen, what happens? There's the damaged cells that produce insulin, as Amna mentioned. Amna, which cells produce the insulin?
The beta cells.
Beta cells. So what happens when the beta cells damages that produce the insulin? It can cause diabetes. So that is all the pathophysiology related to this MCAT question. So you will have a better idea why you are learning at this undergrad level, and so you will correlate clinically when you will practice. Thank you.
Yeah, thank you. So I'll actually be talking, our next question involves the exocrine functions of the pancreas. Yeah, so let's go over this question. During the interval between 30 and 60 minutes in figure 1A, which of the following would be observed in WT mice or wild type mice. So this question also came from the same passage as the previous question. So we didn't need the passage itself, but within the passage we found figure 1A, which I put here. Basically, all we need to do is focus on figure 1A. So this is a graphical analysis question, and let's take a closer look at the graph. So they asked about the WT mice, which is the darker gray with the squares. So we need to focus on just this line. And they also asked between 30 and 60 minutes. So we've just got a little square here where our attention should be going. So first, when we're doing graphical analysis, we've got to look at the x and y intercepts. So the x-intercept here is the time after the glucose injection, and the y-intercept is the level of serum glucose or the level of glucose in the blood. So why I'm emphasizing this is that I remember in my preparation, I've gotten a question or two wrong because I didn't look at the x and y intercepts. I sort of assumed, oh, yeah, glucose, or maybe they're measuring maybe they're measuring insulin, levels of insulin here. Oh, insulin is going down. So these sort of assumptions that I made without getting the full picture, without actually understanding what I was being asked or what they were giving me, these test makers sort of know that students can make sort of assumptions like this, and they make really appealing answers, assuming that you made these assumptions. So that you can't let that trick you. You have to look carefully at everything you're focusing on, what you're examining. So that's why we have to look at the x and y intercepts of the graph. So let me give you some context on the graph because it may be confusing at first. What are these mice and what's going on with glucose injections? So basically, in the passage, scientists injected mice with glucose to observe the blood glucose concentrations in different experimental conditions. So we've got the experimental condition of WT or wild type, which means just a control of mice. And then we've also got the ALB-TG. Don't worry about the context of that, but that's another experimental condition that they did this experiment in. So we're going to hone in on the portion of the graph that they're asking for, 30 to 60 minutes. What do we see here? As time goes on, glucose in the blood decreases at a constant level. That's all we need to basically know. Once we've got that, we can sort of tackle the question and understand the answer choices that we're given. We have to ask ourselves, what's going on here? Why is this going down? And where is this glucose even going? Why is it just decreasing after an injection was given? Where's the glucose going from the blood? So seeing as the level of blood glucose is decreasing, we know that, like from the previous question and from our content review overall, we know that insulin plays a role in decreasing the blood glucose levels. Because if they're too high, they can cause organ damages and different things like that. And we need that glucose. Like if we consume a meal, we're doing it for the sake of energy. So if we consume a meal and the glucose just circulates in our blood, doesn't actually go in our cells and doesn't give us energy, then what was the point of the meal? So that's sort of why glucose is decreasing here. Glucose in the blood is decreasing, but it's not just like excreting from the body or just turning to dust. Glucose in the blood is decreasing because insulin is forcing glucose to enter cells to give us energy. So let's take a look at how insulin works. It's just a diagram of what I said. So insulin serves as basically the key to unlock this glucose channel in our cells. So insulin binds to a receptor on the outside, allows glucose to flow in from the blood. Pretend this white space is the blood and the glucose is decreasing. That's why we see that decrease in the graph. And so, yeah, glucose enters the cells, and what happens here? They can be stored as glycogen, which is our storage form of glucose, and they can also be used for various metabolism and other sort of small, intricate processes that glucose helps to intervene with. So now that we've got the background, we can get back to the question. So blood glucose levels are normally maintained within a normal range because, again, like I said, organ damages, stuff like that, insulin plays a key role in preventing these kinds of things. So insulin decreases blood glucose concentration. That's what you need to know. Like if you're taking anything from all this I'm saying, insulin decreases the level of blood glucose, and glucagon does the opposite, increases the level of blood glucose. So basically, the what insulin does is it promotes glucose reuptake into the cells, into the tissues, and it decreases the glucose production in the liver. The liver also produces glucose through gluconeogenesis. So when insulin is activated, it increases glycolysis and decreases gluconeogenesis because we don't need more glucose. We've already got plenty circulating in the blood. We need to deal with what we've got, and then we'll continue to make more later. Insulin also stimulates glycogen synthesis. Glycogen, again, as I said, it's the storage form of glucose within our tissues, in our liver and muscle specifically. Also, insulin helps with triglyceride, like fatty acids synthesis, and protein synthesis. So insulin helps us a lot by just bringing in the glucose from our blood. So low blood glucose stimulates glucagon release. If we have low blood glucose, we've all had that feeling of, extreme hunger. Glucagon acts in that case to increase the levels of blood glucose and allow it to enter the blood to circulate around. So figure 1A here, it shows that blood glucose levels decrease between 30 and 60 minutes after the glucose injection in wild-type mice. And this occurred because the glucose injection raised blood glucose levels and it triggered the insulin to act to lower that blood glucose. So what can we say here? Let's go through the answer choices. A, it increased glucagon release. Didn't we say that insulin is released and glucagon is the opposite of insulin? So I think we can rule out letter A because it's the opposite of what is actually happening. B, increase plasma amino acids. It actually decreases plasma amino acids because amino acids are used to build proteins. So amino acids are the building blocks of proteins, and we said that insulin promotes protein synthesis. So we wouldn't have a bunch of free-floating blocks if we have the engineered insulin to bring these blocks together and make proteins, right? So we would actually have a decreased level of plasma, or plasma sort of mean blood, amino acids. So that we can rule out B as well. So letter C, increased hepatic glycogen storage, stores. So hepatic means related to the liver. We stated that. And glycogen, as we said, it's the storage form of glucose. So I think this answer choice looks pretty good because once we promote glucose uptake into the tissues, we also promote glycogen synthesis to store that glucose for later. So C looks pretty good, and then D, increased hepatic gluconeogenesis. We already said that gluconeogenesis would decrease in this case because we've already have an abundance of glucose. So the right answer in this case is D. All right, that was a lot, but I'm sure there's just a few key points that you need to know from your content review that will help you apply your knowledge to any sort of question. Once you break down what the question is asking you, sort of hone in on the details that you need to focus on, you'll be able to answer any question.
All right. So you said correct answer is C or D?
It's C.
C. Okay, good. So, so increased levels of insulin cause blood glucose to decrease what? What happens to the glucagon? Increase or decrease? Glucagon release increases or decreases?
Glucagon release would decrease because
Very
they're
good.
opposing processes.
Yes. And what about the amino acids? Increase or decrease?
Amino acids in the blood would decrease.
Okay. What about hepatic gluconeogenesis?
That would also decrease.
Okay. What about hepatic glycogen stores?
Now that would increase.
Yes, good, very good. So what's a medical condition when there's a high levels of glucose and there's a insulin deficiency? What's that condition called?
Diabetes.
Very good. Okay, let's move on.
All right. Okay, so next question is also related to biological and biochemical foundations. So BB, let's go on to the question. So, although the pancreas is located posterior to the peritoneal cavity, pancreatic ascites is a condition in which the pancreatic secretions leak into the peritoneum. The best explanation for this condition would be leakage of what? So there's a lot of big words here, and you'll be familiar with all of these once you've gone through your content review. The GI system likes to be tested a lot on the MCAT. The GI system is sort of a middle yield topic, but it's really important if you just want to make sure that you understand everything, go from head to toe. So we've got to hit the GI in your content review. So let's see. This is another pseudo-discrete question. So it came with the passage, but again, you can see I didn't include the passage because it's not necessarily important. It's just sort of just testing your knowledge. I think the first thing we can do here strategy-wise to just break things down and make it easier for us to get to the right answer we should notice the difference between A and B and then C and D. So A and B here, they mentioned endocrine pancreas hormones. They both start with endocrine pancreas hormones, while C and D include pancreatic enzymes. So let's see here. So the endocrine pancreas hormones include the what we talked about, insulin, glucagon, somatostatin, because pancreas has two functions, endocrine and exocrine. Exocrine includes the pancreatic enzymes. So, but we know that the endocrine hormones, the function of endocrine hormones in general, not even just related to the pancreas, these hormones are circulated throughout the blood. But the question stem mentions that the pancreatic secretions that we're focusing on here, they leak into the peritoneum. The peritoneum is a thin membrane that lines the abdominal cavity and covers most of the abdominal organs, so stuff to do just within the abdomen, within the little cavity of the abdomen. And this, the peritoneum provides like support and reduces friction overall, just supports all the organs. So we know that in this case, we won't actually be concerned about the endocrine function of the pancreas. We're talking mostly about the exocrine function, how how the pancreas excretes these enzymes or these juices, you could say, just within the peritoneum to help with digestion. So we're not focused on endocrine at this time. So we can actually rule out A and B. So to help us continue, let's take a look at how the pancreas is situated in sort of the grand scheme of things. So I actually really enjoyed looking at this diagram is from UWorld because I never actually knew what the pancreas looked like and where it was situated in the whole digestive system, because I thought it was a whole separate organ somewhere else, like perhaps near the kidneys. But I was wrong. Once I got through the content review, I understood. So basically here we can see that the pancreas is situated adjacent to the duodenum. The duodenum is the first segment of the small intestine. So the small intestine has three segments, the duodenum, the jejunum, and the ileum. And then the large intestine also has three segments, the cecum, colon, and rectum. So we see here that normally, let's see, in a healthy pancreas, the pancreatic enzymes, so this yellow thing is the pancreas, not this. This is the duodenum, the small intestine. So we see here that in normal conditions, the pancreas releases these digestive enzymes directly into the small intestine. And this is through the pancreatic duct here. So this sort of facilitates the transfer of the pancreatic enzymes directly into the digestive system. So what are these pancreatic enzymes for? They're to help digest lipids, mostly. So these juices go into the small intestine and to help with food processing and stuff like that. But abnormally, so this is the normal situation, but abnormally, the pancreatic enzymes, we can see that they're leaking. They're not going directly into the small intestine. They're sort of leaking into the peritoneum. It's not very clear here. It sort of looks like they're still going into the small intestine, but it's telling us that it's leaking out away from the small intestine just into the cavity. And that's not good because if they're not in where they're supposed to be, they could cause further problems. They leak into the peritoneum. So let's go back to the question. Now we're between C and D. As we said, we can be endocrine. We're focused on here. We're focused on the exocrine, or the pancreatic enzymes. So we established that the pancreatic enzymes are released into the pancreatic duct, and these enzymes don't go directly into the bloodstream like how endocrine hormones are secreted. So what do we see in letter D here? Pancreatic enzymes, as they are delivered to the blood, supplying the digestive system. But we already discussed that the endocrine hormones go directly into the blood. The exocrine sort of go directly where they're intended to go, which is the small intestine. So we can see between C and D here, C would be the correct answer. Dr. Chimo, would you like to pitch in on this case?
Yeah, I think very good. So whenever there's any leak of in the pancreatic enzymes, pancreatic secretions in the peritoneal cavity, it can cause peritonitis. What's peritonitis? Inflammation of the peritoneal cavity, the peritoneum, the lining, the covering of the GI tract. So I will not go in much detail. Let's move on to next.
All right, so question number 5, let's take a break from the bio and biochem. Let's transition into a little sneak peek of the chemistry physics to take a break from the bio stuff. It can be a lot, but I enjoy chemistry physics because it's more math based. So it's more of the problem solving stuff. Okay, So this is another pseudo-discrete question. Let me read it. Which of the following statements most accurately describes the component that remains in the reaction flask during the steam distillation? So let me just give you the context. Once you've read the passage, you sort of understand the context of what's going on here, who's doing the steam distillation, all this stuff. But basically, the whole passage was on how scientists got together and made acetaminophen, or as we know, Tylenol, through a bunch of small reactions, sort of building upon each other to get to the final product of acetaminophen. So we can logic through this question, assuming we know the fundamentals behind the problem. I've got the structures here that they've mentioned in the answer key, in the answer choices. So here are the two structures, the 2-nitrophenol and the 4-nitrophenol, that we need to sort of deep dive and understand what's going on. So what is a steam distillation, first of all? Distillation is a purification technique in organic chemistry and chemistry overall. It's used to separate molecules based on their boiling points. So a liquid mixture is heated to a temperature that overcomes intermolecular forces. So what's holding if you have a water, for example, how are these water molecules held together? Through intermolecular forces that keep them together. And once you've heated them up to their boiling point, that's when these intermolecular forces can't handle it anymore and they start to dissociate from each other, and that's how we get gas, vapors. So the vapors, in distillation, we sort of extract the vapors of the portion of the mixture that has a lower boiling point. So we extract that first because it boils up first, and then we condense it into a liquid form by cooling it. So we sort of heat it up and then we cool it back down to separate two separate components of a mixture via their boiling point. So a molecule that participates in stronger intermolecular forces will have a higher boiling point. So that's just something that you need to know. Something with more intermolecular forces, we can think about it logically. It makes sense that if you're sort of held tightly together, it's harder to break things apart and harder to get things into the gaseous phase. So hydrogen bonding is a form of intermolecular force, and it can also be intramolecular, as you can see here. This is our 2-nitrophenol, and this is our 4-nitrophenol. So in the 2-nitrophenol, we've got our hydrogen bonding atoms, which is the O's and the N's and the F's in some cases. They are sort of already taking part in their own intramolecular hydrogen bonding within its own molecule. So it doesn't actually interact with other molecules because it's already caught up in its own hydrogen bond within itself. So that means that it has less intermolecular hydrogen bonding, so less intermolecular forces, which means it would have a lower boiling point. So because it'll be easier to dissociate these molecules from each other. All right, so we can see that between choices A and C, 2-nitrophenol, we said that oh, sorry, let me backtrack. So the question is asking that what remains in the reaction flask. So that means that whichever molecule extracts from the reaction flask first, whatever remains has the higher boiling point because it wasn't boiled first. It's going to be boiled with a higher temperature. So whatever is remaining in the reaction flask, which is what they're asking, has the higher boiling point. So what did we say here? That the 4-nitrophenol has the higher boiling point because it has more intermolecular hydrogen bonds. All right, so also one thing that they like to test you on is how hydrogen bonding correlates with polarity. That's a whole separate topic, but that's something to keep in mind as well. So hydrogen bonding increases boiling point and polarity of a substance. So what did we say that the 4-nitrophenol has the higher boiling point, which means it would remain in the reaction flask. So it's not A or C because it's got to be B or D with the 4-nitrophenol. Let's read the remaining part of the answer choices. So 4-nitrophenol remains in the reaction flask because it has more intermolecular hydrogen bonding. Or 4-nitrophenol remains in the reaction flask because it has less intermolecular hydrogen bonding. But we already established that the more hydrogen bonding you have between different molecules, the higher the boiling point, which means that B is logically the correct answer here. I'll go on to the next case. Okay, Our next question is also related to the Chem Phys part, and it's basically a spin-off of the question I presented before, as it came from the same passage. So normal phase thin layer chromatography, or TLC, was used to monitor the formation of acetaminophen. So just like I said, that the passage was all about how they made acetaminophen, and here we're sort of seeing sort of their experimental tactics and how they were able to identify that their reaction was a success. So according to the TLC plate shown, which spot has an RF value of 0.33 and what compound most likely corresponds to this spot? So this question is sort of asking two separate questions, right? So that actually helps us in our process of elimination, because once we've got the answer to one question, we can sort of rule out two of the answer choices. And yeah, so let me just go into the basics. Thin layer chromatography is a technique to also separate compounds based on their polarity. So what's going on here is that via capillary action, basically they put a paper into a cup filled with a little bit of liquid, and this liquid is nonpolar. And through capillary action, the liquid rises up the paper and it separates compounds based on their polarity. So the compounds that are more polar, they don't rise as much. And the compounds that are less polar, they rise with the liquid that goes up, because the liquid was, in fact, nonpolar. So that helps us a little bit. So we can see that the spot of A would be more nonpolar because it's higher than the spot of B. All right. So what is this RF value? RF value is called the retention factor. So it basically tells us how high the dots have gone up the paper. And the RF value is always less than 1. So how do we calculate this? We just divide. It's pretty simple. You just divide how far up the spot has gone divided by how far up the liquid has gone. So for B, the RF value here would be 3 divided by 9, which is 1/3 or 0.33. And for A, the RF value would be 6 because it went up 6 inches or 6 centimeters, or however, whatever the scale is, divided by 9, which is how far the solvent or the liquid that I was talking about is how far it's gone. So A has an RF value of 0.66 because 6 divided by 9 is 2/3 or 0.66. So that's why I kind of like the Chem Phys section, because it's a lot of math. So we said that the spot for B would have the RF value of 0.33. So that answers the first question that it's asking us here, because that means that we only have two options now, B and D, because they both say that spot B corresponds to the RF value of 0.33. So now it's asking which of the compounds actually is what the spot is. So we've got two options, acetaminophen or 4-aminophenol, and these compounds were given to us in the passage. Here we can see that their structures are quite similar, but they differ by one key feature. We see that the aromatic ring and the hydroxyl group up here, they're all the same between both of the structures. But what's down here? Over here we have an amine functional group, and over here we have an amide functional group. All right. So now we need to use our knowledge of polarity of different functional groups, as I'm sure you've learned in your chemistry, your basic college chemistry classes, your freshman year chemistry, to figure out which is more polar. All right, so we've got a diagram here. It's kind of small, but it gives you the big picture that the ranking of functional groups in regards to their polarity. So what were the two functional groups that we had? We had this amide group, which is the carbonyl attached to the nitrogen, and then we had an amine group, which is just a nitrogen attached to 3 groups. It doesn't matter what they are. So over here, let's see, the amine is up here, amide is down here, which means the amide group is, in fact, more polar. So that means that acetaminophen as a molecule itself is overall more polar. And we said that the more polar groups in TLC, they stay behind. So they sort of stick to the paper. The paper is more polar. So you know that like dissolves like, in a similar way that the more polar compound sticks to the more polar substance or the more polar surface. So they like sticking to the paper. Non-polar compounds stick to the liquid. So they push up with the liquid, which is non-polar. So we've got the RF. We've already calculated this for both of the spots. B was 0.33, A was 0.66. And then we know that the more polar compound, which was acetaminophen, is down here. And the less polar compound went up here with the solvent. So we can answer this question now. We know that it was either B or D, and now we know that the compound that corresponds to the spot of B is in fact acetaminophen. So we've narrowed down our answer choices to get to the correct answer. And I love using the strike through tool in these cases because you once you answer the first question, you strike through A and C, and then you answer the second question, you finally get to B. So yeah, Dr. Chima, you want to pitch in?
Sure. So, can you tell me, Amna, what is acetaminophen? Can you tell me?
Acetaminophen is a medicine that provides pain relief and
Uh,
fever control.
Good. So, this medicine is available over the counter in pharmacy. What's the common name? We all know.
Tylenol.
Yes, you're right. Yeah, so basically, I will pretty quick go over this Tylenol, acetaminophen mechanism of action. So you will learn in the pharmacology, in the medical school, all the mechanism of action, the side effects. So let's pretty briefly, I will not go in much detail. So acetaminophen basically blocks the pain by inhibiting the synthesis of the prostaglandins. So what are the prostaglandins? So these are basically a natural substance in our body that initiates the inflammation. So acetaminophen reduces fever by, and this is the pain mechanism, as Amna mentioned, two reasons, pain and fever, right? So this is how acetaminophen can relieve the pain. And the acetaminophen reduces the fever by acting on the hypothalamus region of the brain, which regulates the temperature. So these are the mechanisms of action. And basically, it induces the analgesia. Analgesia means the pain inhibited by blocking the prostaglandin synthesis from arachidonic acid by inhibiting the enzymes called COX-1 and COX-2. So basically, it's a weak inhibitor of the synthesis of the prostaglandins. So I will not go in much detail, but this one pretty quick. This is related to the pharmacology that you guys will learn during your medical school. So you will understand all the basics that you have learned during by taking your MCAT exam, by preparing for the MCAT exam, and we have a good base, and then it will be easy for you in the medical school. What's going on and what's the mechanism of action, right? Thank you.
Yeah, thank you, Dr. Chima. So I'll just a few more questions left. We're going back to the biological and biochem. Just a few more questions. So let's read this one. Which of the following sequences accurately describes the pathway of communication between a presynaptic neuron and a postsynaptic neuron? So one thing that I've noticed is that when I pull the questions from your world, a lot of them a lot of them want you to just a lot of them want you to go back into the deep fundamentals of what you know. So the content that you know, your world likes to bring that all up to the front stage. So you've got everything in your mind swirling around, and then your world helps you to combine all these things into one discrete way of like answering questions. Once you answer questions with this knowledge, then it becomes firm in your mind. Not sponsored, by the way. I just really like your world. So, all right. So this is another pseudo discrete question. It did, in fact, come with a passage, even though it's pretty simple. It's just asking about the basic structure of a neuron. It is a pseudo discrete. All right, so let's just go back to the fundamentals, just like your world wants us to do. Neurons are cells that receive and integrate and transmit information in the nervous system, and communication between neurons involves an electrochemical process in which electrical signals or chemical signals they travel from a presynaptic neuron to a postsynaptic neuron. So the synapse is what connects these two neurons together, and what comes before the synapse is called the presynaptic, and what comes after the synapse is the postsynaptic neuron. So an electrical signal in the presynaptic neuron travels down this thin fiber called an axon, and it conducts the signal. It's usually wrapped with myelin that helps insulate the signal and prevent from loss of the signal. And from there, chemical messengers are initiated to be released into the synaptic cleft where they sort of jump over the synapse. They know exactly where they're supposed to be going. They're supposed to hitch on to the dendrites of the receiving neuron or the postsynaptic neuron. And this alters the electrical potential and it sort of carries messages down a pathway. Nerves are composed of plenty of neurons strung together. So the change in electric potential spreads to the soma, and then it just keeps going. It goes soma, axon, dendrite, soma, axon, synapse, dendrite, and then repeats. So how would we answer this question here? Because we know that we could have a bunch of different orders. That's what they're trying to trick us with, is if we have a sort of set order as to what we're looking for, we might get confused if we don't see that right away. Like in my mind, I would say that the signal goes from dendrite, soma, axon, synapse, but other people might say soma, axon, synapse, dendrite. These are all completely correct ways. They're just sort of swapped in a sense that like the orders are slightly different, but they're all the same. We've all got fundamental things. We just need to keep in mind the few things that we need to make sure that our answer choice matches. That axon has to come before synapse, or that dendrite has to come right after synapse, and the soma has to come right after the dendrite. So no matter what order we see it in, it's got to follow these certain patterns. Because at first, when I was answering this question, I was like, wait, I'm thinking of dendrite, soma, axon, synapse, and it's not here. Dendrite, there's no dendrite, soma, axon, synapse. And then I got confused until I broke it down and I sort of drew it out. I was like, oh, you can just reorder them and any order would be fine. So don't get flustered, just break the answer choices down and trust your knowledge. So in A, the axon does not immediately come before the soma because we said the axon has to come before the synapse, so it's not A. B, we see that everything is in a good order, and it was one of the combinations I actually wrote here: axon, synapse, dendrite, soma, axon, synapse, dendrite, soma. So I'd be confident to pick this one and move on. But sometimes it's good to just check the other answer choices. But in a relatively simple question like this, I'd say that to save time and to save time for the more complex questions, you could just pick B and move on. You can flag it if you really, really want to check back, make sure the other answer choices are not right. But again, only one answer choice can be correct, and we know that B is in fact correct. So in this case, we would just move on. So just a couple of more questions, guys. We got this. So this one is another pseudo-discrete, came with the same passage as the one before, but it's just asking stuff that we can pull from our knowledge. Which of the following is true regarding the organization of neuronal tracts in the spinal cord? So here's basically all you need to know for this question, because we're given a couple of keywords: afferent and efferent. That's how we differentiate between the first two answer choices and the last two. And then we also got motor commands versus sensory commands. And then we also have gray matter and white matter. So these are three things that we need to keep in mind and differentiate to make sure that all three facts are in alignment with each other and make sure we pick the absolute factual, objectively correct answer. Because your answer choice shouldn't be partially correct. It's still wrong. So this is all you need to know. The central nervous system, it includes the brain and the spinal cord. It's composed of white matter and gray matter. So that was one of the key terms that we had to differentiate between in the answer choices. So the gray matter is located in the center of the spinal cord, and it's composed of unmyelinated neuronal cell bodies and dendrites. But the white matter is composed of axons, and it's in the periphery of the spinal cord. And these are composed of myelinated and unmyelinated, but mostly it's myelinated because it allows for long distance travel of communication, make sure that signal doesn't get lost. So white matter consists of afferent and efferent axons. So afferent, what's the difference between afferent and efferent? So afferent is ascending. So how can we this is sort of a mnemonic. Afferent is ascending. So afferent takes messages and ascends them up the spinal cord to the brain. It carries sensory information from the body to the brain. Like, for example, if you touch a hot stove or a hot pan, for example, you touch it, and afferent neurons carry that pain, that sensation of pain up the spinal cord, like ascend up the spinal cord to the brain to initiate what comes next. So those are afferent neurons. And then efferent neurons are descending, or I like to think of it as exiting. So they exit the brain and release through the rest of the body. And they carry motor commands. So what are these? In the case of the hot stove example, once you get the signal that you're feeling pain, the efferent neurons command your finger to, jerk back away from the stimulus. So that's the difference between afferent and efferent. So we've hit white matter, gray matter. We've hit afferent, efferent, and now we need to hit the sensory versus motor, which we also hit that. So afferent carries sensory information, efferent carries motor information. So therefore, in the spinal cord, efferent neuronal fibers carry motor commands from the brain to the body through spinal white matter in the periphery of the spinal cord. But on the other hand, afferent neurons carry sensory information from the body to the brain. Okay, so what matches what I just said? The letter C. So we can go through all of them. So choice A, afferent neuronal fibers carry motor commands from the brain to the body through tracts in the spinal gray matter. So what did we say? We said that afferent neuronal fibers ascend the spinal cord to the brain. So they carry sensory commands, so not motor commands, that's already wrong, from the body to the brain, not vice versa. So A is wrong through process of elimination. B is also wrong because afferent neuronal fibers, they do carry sensory information, so that's correct. From the body to the brain, that's correct, through tracts in the spinal gray matter. But that part here is what is incorrect here. So, test makers like to trick us in that, oh, the first like plenty of words seem perfectly correct. We might pick B and move on as an impulse to try to get through questions fast. But we have to notice these last two words completely nullified the whole entire answer choice. So that's why we have to eliminate that and move on to the next choice, which is efferent neuronal fibers carry motor commands. Check. From the brain to the body, check. Through tracts in the spinal white matter, check. So all three of those components of that answer choice are completely correct. You can check D if you want. So we see that efferent carries motor, not sensory. So we know that D is wrong because any answer choice that is only partially correct is still absolutely wrong. All right, so one more question related to the sciences. Then we'll go into some psych-soc practice. Which of the following is true regarding the functions of neuroglia in the central and peripheral nervous systems? So we've got a couple of answer choices here. First, we need to have that background information. Like I said, content review is the key thing that'll help you in the long run. Once you've got a solid foundation of content, you can apply that to any question using test strategies. So what are glial cells? This is another, a content review question. We need to take back to what we need to know to answer this question because it's asking a lot of questions with like buzzwords and these vocabulary terms that we'll not be familiar with if we don't go over the vocabulary that we need to know. So the nervous system, let's talk about what are glial cells. Then we'll talk about the sort of building blocks or the different types of glial cells. The nervous system is composed of both neurons, which are, nerve cells, and glial cells. Neurons conduct electrical impulses, and glial cells serve a variety of vital support functions that support the health and nourishment and nutrition and overall function of the brain. So astrocytes, these are things that, let me get into the definitions first. So astrocytes nourish neurons and form the blood-brain barrier. So that means that nutrients in the blood can get through the brain via astrocytes. Ependymal cells over here, they line the ventricles. You can see they're sort of in that columnar, sort of similar to like endothelial cells. They align our organs. Ependymal cells line the ventricles of the brain and produce cerebrospinal fluid to help nourish the brain as well. And oligodendrocytes and Schwann cells. These both actually produce myelin, and myelin is used around axons, like I said, to help propagate the signal and make sure we don't lose any of that signal. So, but what's the difference? They both seem to do the same thing. But the difference is that oligodendrocytes are found in the central nervous system, while Schwann cells are found in the peripheral nervous system. I have a mnemonic for this. It's COPS, so COPS. Central oligodendrocytes, CO, and then peripheral Schwann cells, PS. So that's how you can differentiate where you find the two. And finally, we have our microglial cells, our microglia. These are phagocytic and they help to get rid of waste in the central nervous system. So again, this stuff At first, I was having a lot of trouble in remembering all this because it's like just five random names of random cells. How am I supposed to know the functions and where even I find these? So what helped me was looking at pictures. So I really like this picture. It's really colorful. It helped me remember where everything goes. You can see astrocytes hugging the blood vessel. Microglia sort of look like little phagocytic little monsters. So through images, I was able to memorize, but also the most important thing that helped me was writing it down. So even writing it down once or twice was just helped me sort of recall that as I was doing questions, I was able to imagine, oh, I wrote that down on this page in this notebook in this color. So I remember the content of that information. So just writing things down and looking at images, doing what's works best for you, make sure that you stick with that and it'll all work out. So in this case, we'll go through each answer choice. Astrocytes are peripheral nervous system cells that regulate neurotransmitter concentrations. So first of all, astrocytes are central nervous system cells, and nourish they nourish the brain. They don't exactly regulate neurotransmitter concentrations. That's not exactly one of their functions specifically. So confident. I would be relatively confident in striking out answer A. If you're not completely sure, you can save it for until you see something that you're 100% like, oh, this must be the answer. So letter B, each Schwann cell myelinates multiple axons in the central nervous system. But we said here, the difference between oligodendrocytes and Schwann cells are the fact that oligodendrocytes are the central nervous system and Schwann cells are peripheral. So this says that Schwann cells are in the central, which is wrong. So we can rule out B because we know that Schwann cells do myelinate axons, but even the fact that this word right here, central, is what makes this whole answer choice incorrect. Now answer choice C. Each oligodendrocyte myelinates a single axon in the peripheral nervous system. Again, they swapped the roles. This should be central, then it could possibly be correct. Although the single axon thing might be questionable as well, but we know for sure that this is wrong because it says peripheral nervous system. But letter D, microglia act as the macrophages of the central nervous system and consume waste. That is exactly the definition that I stated, that I have in my knowledge bank in my mind. That completely matches, so I would be confident in ruling out answer A for sure and going with D. Yeah. So overall, the educational, like what was the point of this question? It's just to get you to know the supporting cells of the nervous system. What goes on in the brain besides just neurons carrying signals? How does the brain actually function and nourish itself and maintain its own health? Okay, so our last question. I know we had a lot of fun. It's been an hour. So our last question is related to the psychological, social, and biological foundations of behavior. We like to abbreviate this to just call it the PS section or psych-soc. And I find this section actually the most fascinating because it's much easier to apply concepts of sociology and psychology that you learn through Anki or through book reviews and reading that 300-page doc that's so famous. So it's much easier to apply these concepts into your everyday life right now, although you might be able to apply your biological, your chemical, all that stuff can be applied in the future. But for now, in the here and now, as we're not physicians yet, we can only apply these psych-soc concepts to our daily life to able to understand them better. And it's easier to understand things when you make personal connections. So let's go into the question. Um, I have a question here that let me read it for you. Which of the following illustrates a central route to attitude change? So a person is influenced by a doctor's advice because the doctor has Ivy League degrees. A person is influenced by a doctor's advice because the doctor's speech is highly eloquent. C, a person is influenced by a doctor's advice because the advice is clear and evidence-based. Or D, a person is influenced by a doctor's advice because a website calls the doctor a top expert. So we see here that all these answer choices are pretty similar. The first couple of words are all exactly the same, but the reason why the doctor's advice was taken is different. So let's go into the basics of what's going on in this question. So within persuasion, we have two main routes, the central route and the peripheral route. So the central route is where we engage deeply with the argument, and the peripheral route is where persuasion is more about the attractiveness or the superficial nature or the superficial aspects of the message itself. So these two routes of persuasion fall under the umbrella called the elaboration likelihood model. So the MCAT likes to test terms like these that you're able to learn these terms through stuff like Anki that really helped me. So these two routes, we know that the question asked about the central route. Let's go into these two. So let's say, for example, how do we differentiate between the central and peripheral? The best way to do this is by examples. We've got the definitions here, but it's hard to apply these if we don't have specific examples that we can stick in our minds and be like, oh, this situation matches what I have encoded in my mind, so I'll be able to pick this answer choice. So, for example, a central route to persuasion. We could say like a public health campaign promoting vaccination, but they use detailed, specific scientific data and expert analysis, expert testimonials to explain to the public how these vaccines work and the benefits of getting vaccinated. All this stuff, they provide evidence to the audience, which is the public, for why they should get vaccinated. But on the other hand, we've got the peripheral route, which is we could say that we have a commercial for a new smartphone. But in this commercial, the commercial relies on attractive visuals, flashy colors, and maybe like a popular celebrity's endorsement, catchy music, all this superficial stuff that doesn't exactly give us information about like um, the aspects of our cell phone that they're advertising to us. So this would be the peripheral route. So we've got the central and the peripheral. Let's go back to the question. So let's see here. We can see that we're influenced by the doctor's Ivy League degrees. That we could say is more of a superficial trait of the doctor. It doesn't actually give us information about the doctor's advice or why they're giving us this advice. And this influence comes from just the qualifications of someone delivering the message, which is not central, it's peripheral. B is also incorrect because it illustrates a peripheral route, which is the influence comes from the verbal eloquence, which is also an artificial or superficial aspect of persuasion. C is actually correct because it reflects an evaluation of the content and message. It allows for a clear and concise evidence-based argument or conveyance of this message that the doctor wants to give you, and it helps a patient understand the doctor's message more when it's delivered through the central route rather than through these flashy artificial peripheral route. And letter D is also incorrect because a website calls the doctor a top expert, which is also a peripheral route effect. And the influence here, it comes from just the reputation of someone delivering the message, which is similar to letter A. One other strategy I like to share with others is that two answer choices can't be correct. So if you have two answer choices that are basically telling you the same thing or basically hit the same fundamental concepts, they both can't be correct. So they both probably are wrong because you can't have both answer choices. So only do this in cases of when you're very sure that two answer choices are telling you the exact same thing fundamentally, because then you'll be able to eliminate and just be left with the 50-50 if you're completely unsure. So that's all I have to say about persuasion. And Dr. Shima, would you like to discuss personality?
I think you're doing good. You can talk about the ocean. Yeah, please go ahead.
Okay, so let's start the big 5 factors of personality. The MCAT likes to test this occasionally. We've got O for openness, C for conscientiousness, E for extroversion, A for agreeableness, and N for neuroticism. So these are considered the 5 major dimensions of human personality. It's best to think of this as the OCEAN acronym because it helps you, remember the 5 um, big factors, helps you maintain that in your mind, not get mixed up. So keeping these mnemonics, OCEAN, just helps you remember these 5 aspects. So here are some of the details. So this model is also known as the 5-factor model or the OCEAN model again. So the MCAT in this case, like I actually did a question about this a few months ago, that the MCAT asks questions related to this concept. They don't ask like, oh, what is openness? And they give you like a list of traits. They ask questions like, given these specific traits that you witness in this patient or you witness in your classmate or friend, what would this person have a low score on? So let's say like they describe a person who is not open to taking new ideas. They include keywords like that, open. They're not open to feedback, or they stay in one lane throughout life. So what would this person have a low score on? Would be openness. They would also give you options like extroversion, agreeableness, neuroticism, but the answer would be, in that case, it would be openness. So that is all the questions I have for you all today. Thank you so much for saying with me we went through a lot of questions, we went through a lot of details, but it's important to remember that in the MCAT, all you need is a solid foundation of knowledge. And then once you do practice questions, you build up that knowledge, you build up the confidence to apply your knowledge to any situation that you come about. And building up a stamina, so like taking practice questions in large chunks of time or taking full lengths, stimulating testing conditions helps you build up the stamina to ensure that you are ready for test day. Dr.
Right.
Shima, you
Thanks,
can take it
Amna.
away.
Really appreciate. So as you know, any session you guys are with me, I talk about the self-care. So what is self-care? So self-care is basically defined as the process of establishing behaviors to ensure well-being of oneself, to promote health, to actively manage any illness if happens, and and basically it's a practice of individual looking after their own health, using the knowledge, using the information available. And it's basically a decision-making process that we decide that we have to take care of our well-being, our self. And why it's important? It's important because if we are not healthy, if we are not taking care of ourselves, and we cannot give it to other what we don't have. So it's very important to work on our health, our well-being, and self-care. So it's very important for you guys to take care of yourself as well and make a habit to practice taking actions to preserve and improve your health on daily basis. And I all talk about these four main domains about self-care and well-being: the physical health, physical intelligence, intellectual health, emotional health, also called mental health, emotional intelligence, and spiritual health and spiritual intelligence. So physical health, I'll focus on one domain, like table has four legs, right? So we need to work on all four areas, in all four domains, to keep us as a whole well, for our well-being. So physical health, I make a mnemonic for myself as self, SELF, S for sleep hygiene. It's very important. It helps with um, our body health as well. So then exercise. So try to do 5 to 10 minutes every day exercise, and anything that make you move. And then L, learn how to manage stress, and F is for food, add nutritious food. So I will briefly talk about the stress. What is stress? So if we know what is stress, then we are able to manage. So basically, what happens, why we have stress? So it's sometimes it's too much to do, or we have too little time to do. Sometimes we have a low energy level, and that we don't have to do what we don't have time to achieve what we want to achieve. So basically, stress is the gap between our expectations and the reality. So wider the gap, the more stress we feel. So how we can close this gap? So it's very important to, first of all, understand why we have stress, why there's a gap between our expectation and reality. First of all, we have to understand we are making the smart goals. We are setting up smart goals for us that we can achieve in a timely manner. So how we can uh, keep this gap closer? Action, take actions. So whenever there is a misalignment, stress happens. So between misalignment between our desires, our goals. So what are we supposed to do? We have to take actions, right? Like you have goals to take MCAT, right? Your goal is to go to the medical school, right? Your goals to be a physician, right? So we all have our goals, our desires. So we have to make a realistic goals, first of all, to achieve them. And how we can achieve, take actions, how we can take actions, like start reading, right? So like for MCAT exams, it's not just we can cram overnight and go tomorrow and take exams. No, it's take small baby steps, and it will help you to build up, have a strong knowledge base. And then these small steps towards your goals can help alleviate your stress level. And by aligning your actions with your intentions, with your goals, and giving you a sense of purpose and progress. And this is how you can manage your stress on a daily basis, to one day at a time, take small baby steps and see what you can achieve in the given certain time period. All right, so I always talk about the keys to success. Everyone has their own definition of success. And I always say, for me, the one I learned is help ourselves and help others. How we can do that? We have to achieve our goals, right? How we can do that? We have to be passionate about it, whatever our goals are. And we all want to have our professions. Our passion and profession should be the same. We have a same passion and profession. We are not working a single day. And then persistence, as I mentioned, take small baby steps, and perseverance. And there's a trait, a very good personality trait, grit. Grit is basically characterized by the perseverance and passion for achieving the long-term goals. And it entails working strenuously to overcome the challenges, obstacles, maintain your efforts on daily basis, and keep interest over time despite any failure, despite any setbacks, and keep progressing, keep improving your learning curve. And last but not least, practice, practice, practice. There is no magic in this world. Everything is practice. So if you don't remember, like prep cycle or like MCAT question, so keep doing it over and over again, second time, third time, fourth time, fifth time. So just make sure, I understand it, we feel like, oh, who can do the 4 or 5 times, right? But this is how we can make the tracks in our brain to understand the concept and memorize them. And then everything is practice in this world. So I will stop here. I wish you all best of luck. If you guys have any questions, please feel free to send us the email, and we'll try our best to answer and try our best to help as much as we can. And definitely please start helping others as well. All right, I'll stop here. Thank you, everyone.
Yeah, thank you so much, Dr. Shima. And again, if you guys need any advice or would like to discuss anything further, please feel free to email us at learn@teleshadowing.com. And the quiz is now live on the website. So again, a 70% or higher to pass and receive certification. And yeah, thank you so much to everyone for tuning in today. Hope to see you at our upcoming sessions as well. Have a great day.